Uh, Today we shall discuss about all are non-principal. Uh, Threshold and all are non-response. What do we mean by threshold and all are non-response? As we have discussed in previous slides, that not all the membrane depolarization in excitable cells they trigger the positive feedback relationship and ultimately production of an action potential. I mean, action potential is not always generated in all of the membrane, even uh, if they are excitable membrane. So event that initiate the membrane depolarization, it basically provides an ionic current. I have told you that for the depolarization or for the production of action potential, there are two factors. One is concentration gradient, as a result of which the ion will move either from outside to inside or from inside to outside. And there is another electric potential which may operate in the opposite direction. So ultimately there will be the initial depolarization or generation of the potential from the resting membrane potential, which is called depolarization of the membrane. So as the depolarization begins, as I have told you, that potassium will start to move out from the leaky potassium channel, and it will efflux of the potassium will increase above its resting state, because inside negativity it ultimately it tend to keep the potassium in the cell that is weaker as compared to the outer side positivity. So as all of you know that like charges they repel each other. So positive charges they will start to repel the positive charges, and negative charges they will start to repel the negative charges. Whereas in case of opposite charges, an electric force will draw those opposite substances together, and separated electric charge of the opposite sign, they have the potential, they have the power to do work. Mean, if the one side of the membrane is having the positive charge and other side is having negative charge, it means that membrane is having the potential to do work, because that membrane is having the potential difference, charge separation, and those charges are of opposite nature. And when they are allowed to come together, they will result in any type of the work. So this potential is called electric potential, and because it is determined by the difference in the amount of the charge between 2 points, a potential difference, we can also call it as the simply we can also call it as the potential. So when any stimulus is applied to membrane, initial movement of the sodium into the cell will decrease because of the same lessened negativity. However, when there is depolarization, as I have told you, that voltage gated sodium channel open, and sodium permeability will be increased, and ultimately sodium influx will also enhance. And all in all, at this stage, potassium exit still exceed sodium entry, because I have told you that potassium channel they open later, quite later as compared to the sodium channel, but the potassium channel they remain open even after the closure of the sodium channel. So once a new action potential or new depolarization event has been initiated on the plasma membrane, the potassium channel still they are in open phase. And when the potassium channel are open, as all of you know that potassium is having higher concentration inside the membrane. So hello. Hello. So potassium will tend to leak out from those open potassium channel, and when the stimulus will increase, or stimulus continues to add the current or the positive charges to inside of the cell, the depolarization will increase, and more voltage gated sodium channel will open. Now with the passage of the time, or with the application of the stimulus, and when the intensity of the stimulus is increased, sodium channel will again they will open, and sodium permeability will also open, as a result of which sodium influx will increase as compared to the potassium efflux. I mean, potassium will continue to move outside, from inside of the cell, and sodium after opening of the voltage gated sodium channel, now the sodium influx will increase, and it will be more than the potassium efflux. Once the point is reached that the sodium influx, it will exceed the potassium efflux, the positive feedback cycle take off, and electric potential or the action potential will be generated on the membrane. From this moment, now the membrane events are independent of the initial disturbing events, and now they are totally driven by the membrane properties. Now it means that once the action potential has been generated by any stimulus as a result of opening of the sodium voltage gated sodium channel, and sodium decreased as compared to the potassium efflux. So as a result of that, action potential has been generated on the plasma membrane. Now the membrane events, they are not dependent on the intensity of the stimulus in case of action potential, because once the action potential has been generated, it will be transferred all along the membrane with same intensity, or unanimously it will be transferred all along the membrane. So action potential occurs only when the net movement of the positive charges through ion channel is inward, means net movement is towards the inside of the membrane. So membrane potential at which this occurs, or the action potential is generated, that potential is also called as threshold potential, and the stimuli that are strong enough to depolarize the membrane to threshold level are called threshold stimuli. So it means that the stimulus which is capable of moving the membrane potential from the resting membrane potential to threshold level, those are called threshold stimuli. So for example, this is the threshold level. Threshold level mean the value of the threshold potential is plus 55. It means it will go from minus 70 to 0, and value of the threshold potential is minus 55. So it will go from minus 70 to minus 55. When it will reach the minus 55 value, this is the threshold value. Mean when this value is attained by a membrane, now this membrane will be able to generate an action potential, and all the potential which are below this threshold value or the threshold level, those will be called as subthreshold potential, and the potential which is capable of generating the action potential, that is called threshold potential. So once the potential reaches to this point, now the membrane potential will be independent of this initial intensity of the stimuli. Now here you can see that all along the membrane potential is generated with same intensity and unanimously. This is called action potential. This is the property of the action potential, that action potential is actually it is propagated along the length of a membrane in non-decremental fashion. Mean once it has attained the threshold value, now it will be unanimously generated all over the surface of the membrane. So this is called, and the stimuli which are capable of generating the membrane potential, those are called threshold stimuli. So once this threshold stimuli is reached, it will result in generation of the action potential. So changes in the membrane potential with increasing strength of the depolarizing stimulus, and the membrane potential when it reaches to threshold value, as I have told you that threshold value is minus 55 millivolt, action potential are generated, and when we will increase the stimulus strength above the threshold value, it will not have any effect on the action potential, on the intensity of the action potential. When after hyperpolarization has been omitted from this figure, and the absolute value of the threshold is not indicated, because it can be variable from cell to cell, but generally the value of the threshold is minus 55. So threshold of the most of the excitable membrane is about 15 millivolt less negative than the resting membrane potential. It means the threshold value in most of the excitable membrane is 55 millivolt. So if the resting potential of a neuron is minus 70 millivolt, the threshold potential will be minus 55 millivolt. This is not plus, it will be minus 55 millivolt. And at depolarization less than threshold, outward potassium movement will still exceed the sodium entry, and the positive feedback cycle cannot get start despite increase in the sodium entry. So in such case, the membrane will return to its resting level as soon as the stimulus is removed. So it means for the generation of the action potential, the membrane potential initially it should reach up to the threshold potential. So weak depolarization are called as subthreshold potential, and the stimulus that causes them, they are called subthreshold stimuli. Here you can see if the subthreshold potential are generated, they will not be able to generate the action potential, and the stimuli which are capable of generating these subthreshold potential, those are called the subthreshold stimuli. So the action potential stimulus of more than threshold magnitude, they will also result in generation of the action potential. But as we have seen in the previous slide, in the figure, that action potential resulting from such stimuli, they will have same amplitude as those caused by the threshold stimuli. So we only require the threshold stimuli. Even if the stimuli is of the greater strength than that of the threshold, it will still have the same effect as that of the threshold stimuli. So it means that once threshold is reached, the membrane events, they are no longer dependent upon the stimulus strength. Now the action potential will be generated by the membrane itself. Now it is not dependent upon the stimulus strength. So the depolarization generates an action potential because the positive feedback cycle is operating. So in this case, the sodium influx will exceed than the potassium efflux, and positive feedback cycle will be operating. So action potential, it will be generated either maximally or it will not occur at all. Another way, this is what we can call it as the action potential or all or none. Mean either the action potential will be generated if the stimulus is of the threshold intensity, the action potential will be generated, or it will not be generated at all. It means that the stimuli are of sub-threshold amplitude or sub-threshold intensity. So the actual shape and amplitude of the action potential, it depends upon the membrane condition which are existing at a given time. For example, if the extracellular sodium concentration changes, the shape of the action potential will also change. And this all or none principle is very much similar to that of firing of a bullet from a gun. It means that firing of a gun is a mechanical analogy, and that is also showing the principle of all or none behavior. I mean the magnitude of the explosion and the velocity at which the bullet leaves the gun, it will not depend on how hard the trigger is squeezed. I mean, it is not dependent upon the squeezing or the intensity of the trigger squeezing. Either the trigger is pulled hard enough to fire the gun or it is not. Mean either the gun will fire the bullet or not, and it is not dependent upon the intensity of the ट्रिगर स्क्वीजिंग, यानी आप जोर से बंदूक का ट्रिगर दबाएंगे तो उससे कोई धमाका जो है वो ज़्यादा जोर का नहीं होगा, एक्सप्लोजन ज़्यादा जोर का नहीं होगा. So it is very much analogous to that of the generation of the action potential. So this is called all or none principle. Because of its all or none nature, single action potential, it can't convey the information about the magnitude of the stimulus that has been initiated. Mean from the action potential we cannot estimate the intensity or magnitude of the stimulus, that how hard was that stimulus. For example, how one can distinguish between a loud noise and a whisper, a light touch and a pinch. This information, it will depend upon the number and patterns of the action potential which are transmitted per unit of time, and that are not dependent upon the their intensity or their magnitude. There is another term that is also used while we are studying the action potential generation, that is called refractory periods. During the action potential, a second stimulus, no matter how strong, it will not produce a second action potential, and the membrane is said to be in its absolute refractory period. This is not the case with the graded potential, as we have studied that during the graded potential, you can even before dying of the first action potential, you can generate a second action potential as well. But if we look at the this action potential, in case of action potential, you can't apply a second stimulus once the threshold value has been achieved by the first stimuli and the action potential has been generated. This is called as absolute refractory period, and this occurs because the voltage gated sodium channel, they enter a closed inactive state at the peak of the action potential. I mean, once the action potential has been generated, voltage gated sodium channel, they have been open, and once that action potential has been generated, now these voltage gated sodium channel, they will start to close, and once they are closed, they will remain in inactive state for a shorter period of time. So at that time, if you apply another stimulus, that will have no effect on the voltage gated sodium channel, and voltage gated sodium channel will not either respond to that stimuli. So membrane must repolarize before the sodium channel proteins return to the state in which they can be opened again by the depolarization. So if the depolarization on a specific segment of the membrane has been achieved and the action potential has been generated, that area can't be stimulated by another stimulus unless until that area again reaches to the repolarization state, or the action potential has actually passed over that area, and the sodium channel, they have been they have been closed, totally closed, and they are ready for application of another stimuli. So this is called absolute refractive period. And there is another interval, there is an interval during which a second action potential can be produced, but only if the stimulus strength is considerably greater than, excuse me, we call it in. So there is another condition in which or an interval during which a second stimuli can be applied, but the condition is that the strength of the second stimuli, that should be greater than the first one. In that case, the second action potential can be generated. This will be called as a relative refractory period. So we have two types of the refractory period. One is called absolute refractory period, in which the membrane potential can't be generated even we apply a stimulus of the greater strength. But in second case, we have an interval in which is by the application of second stimuli, the membrane potential can be generated. This is called relative refractory period, and it only lasts for 10 to 15 milliseconds or longer in neurons, and it sometime it can coincides roughly with the period of after hyperpolarization. So during the relative refractory period, there is lingering inactivation of the voltage gated sodium channel, and increased number of potassium channel are open. Mean at that time, this is very short period. At that time, the sodium channels, they are in a phase of in a closure phase. They are entering in a closure phase, but still they are not closed. So due to that lingering, due to that slow process of the closing of the sodium channel, we have a very short interval, very short period at which a second stimulus can be applied. This is called relative refractive period. And if depolarization exceed the increased threshold or outlast the relative refractive period, additional action potential that will be fired or that will be generated. So the refractory period, they basically limit the number of the action potential that can produce by an excitable membrane in a given period of time. So they also increase the reliability of the neuronal signaling, because they help to prevent the production of extra impulses. So most nerve cells, if you talk about the different nerve cells, they respond at frequencies of 100 action potential per second. Mean most nerve cells, they can produce 100 action potentials per second, and some they may even produce much more frequencies for brief periods. And the refractory period, they are very much important, as I have told you that they are important in limiting the number of the action potential generated by a membrane in a unit time, as well as the refractory periods. They are also a key factor in determining the action of the action potential propagation, mean in which direction the action potential is going to be propagated. This is another important factor. So action potential propagation, that how action potential is generated along the membrane, as inside of the cell become positive with respect to the outside when the action potential is generated. So that area of the membrane where the action potential is generated after application of the stimulus, now it will be positive with respect to other region where the membrane is still at the resting potential phase. So the difference in the potential between the active and resting region, they will cause the ion to flow, and the local current, it will depolarize the membrane adjacent to the action potential side to its threshold potential, and sodium positive feedback cycle, it takes over and new action potential occurs there. So this is actual mechanism that how the action potential is generated. So here you can see that this is the site of the present action potential. When the action potential is generated, sodium channel will open, as a result of which sodium ion will move inside, and inside will become positive and outside will become negative. So this area is basically depolarizing area, but the adjacent areas, this is in the resting phase, as well as this is also in the resting phase. So when the action potential is generated, now you can see that here are the negative charges and here are the positive charges. So these, now the positive charges which are present at the adjacent area, they will start to move towards the negative charges, and when the positive charges, they will move towards this area, now the action potential will also be generated to this area as well as this. So here you can see that how this is the direction of the propagation, that action potential will actually be propagated in this direction. So this area is present site of the action potential. This is the intracellular fluid. This is the repolarized membrane, mean it has restored to the resting membrane condition. This is again the repolarized membrane, and this is in the relative refractory period, and this area is representing the depolarized area. Depolarized area mean now the action potential is actually at this specific area, which is called depolarized, and this is polarized but soon to be depolarized by local current. Now this area is still, it is in the resting phase, but soon as the current will propagate to this area, the action potential will also be generated to this area. So it means that there is continuous cycle of repolarization and depolarization. Mean initially this area will be depolarized, and this area will be in the resting phase. Once the action potential has passed over this area will again be in the repolarized condition, and this area will be depolarized condition. When the action potential will pass from this area to this area, it will be converted into the repolarized area, and this will be called as depolarized area. So this is the propagation, or the direction of the propagation of the action potential, that how action potential will be generated. So the action potential that will produce a local current of its own, it will depolarize the adjacent area and it will produce another action potential to the next side. That's why I have told you that once the threshold value has been achieved by the membrane, now rest of the area of the membrane will produce action potential by its own property. So here you can see that once the action potential has been generated, this action potential will produce a local current of its ion, where positive ion will move towards the negative ion, and when the positive ion will move towards the negative ion, the adjacent area will again be depolarized, and there will be excess of the negative charges. And from that area, from that depolarized area, the positive ion will again move towards the adjacent area and they will depolarize the adjacent area. So hence the action potential will be generated all along the length of the membrane. And there is sequential opening and closing of the sodium and potassium channel along the membrane. As all of you know that where the membrane is depolarizing, that area will have the opening of the voltage gated sodium channel, and the area where the membrane is repolarizing, that area will have the closed sodium channel, and that they will have potassium channel that will be still in the open or in the leaky condition. So it is basically खरीद आ जाए जी जी ठीक. It is like lightning a trail of gunpowder. Mean the action potential, it does not move, but it sets off a new action potential in the region of the axon or the area which is just ahead of this particular area. So each action potential, it is as I have told you that it is dependent upon the sodium feedback cycle of the membrane, where the action potential is being generated. The action potential, when it arrives at the end of the membrane, it is identical to that is present at the initial segment of the membrane. That is what we can call it as the all or none principle, that either the action potential will be generated or it will not be generated at all. And once the action potential is generated, it will be same all along the length of the membrane. So action potential are not conducted in decremental fashion as like that of the gradient potential. And the membrane areas that have just undergone an action potential, they are refractory and cannot immediately undergo an action potential. And direction of the action potential, as I have told you, that it is away from a region of the membrane that has recently been activated by the stimulus. So if the membrane through which the action potential is traveling, if it is not refractory, it means that membrane will be capable of producing the action potential in either direction. Mean the direction of the propagation that will be determined by the stimulus location. For example, the action potential in the skeletal muscle cell, they are initiated near the middle of the cylindrical cells and they are propagated towards the end. So if the membrane is not the refractory, so it means the action potential can be propagated in bidirectionally or in two directions. In most nerve cells, because the action potential are initiated physiologically at one end of the cell, mean it will be generated at the axon area at the cell body and it will be propagated towards the other end. So in case of nerve cells, the action potential will propagate in a single direction, but when we look at the certain muscle cells, the action potential can be generated in the bidirectional way. So the propagation ceases when the action potential reaches the end of the axon. I mean, when the action potential reaches at the end of the axon, now it will not be further propagated, and velocity with which the action potential propagate, it is dependent upon certain factors. One important factor is fiber diameter, and the second important factor is whether the fiber is myelinated or it is unmyelinated. So if the fiber diameter is large, it means the action potential will be propagated in more faster way, because it will have more area for traveling. So that's why larger the diameter, fiber diameter, greater will be the action potential, or more quickly the action potential will propagate along the length of an axon or a nerve cell. So large fiber, they will also offer less resistance to the local current, and more ions will flow in a given time, and these ionic movement will break the adjacent region of the membrane to the threshold very quickly. Myelin, as I have told you in a very first lecture of this chapter, that myelin is basically an insulator, and it helps in fast propagation of the current along the membrane. So it So it makes it more difficult for charge to flow between intracellular and extracellular uh, fluid compartments. So there is less leakage of the charge across the membrane. The gradient potential, it can spread along the axon, and concentration of the voltage gated sodium channel in the myelinated axon, or the myelinated region of the axon, is low. So the action potential, so it means that where the myelin is present, myelin will prevent the leakage of the current, so the action potential will not be generated at the myelinated area. Rather, and myelinated area will also have very less number of the voltage gated sodium channel. So as all of you know, that the action potential will be generated at specific area in case of myelinated neurons, and those areas are called nodes of Ranvier, because myelin coating is interrupted or absent there, and the concentration of the voltage gated sodium channel will be high. So it means that fiber diameter, it directly affects the action potential propagation, and myelination of the neuron, it inversely affects the propagation of the action potential along a neuron. So the action potential, it will literally jump from one node to the next, and it will propagate along the myelinated fiber, and that conduction is also called as saltatory conduction. It means salty, or in a leaping manner, the action potential will be conducted along the myelinated fiber. So propagation via saltatory conduction is faster than propagation in non-myelinated fibers of the same axon, because in case of non-myelinated fiber, there will be the leakage, and charges will leak through the myelin covered section of the membrane. More charge arrives at the node adjacent to the active node, and an action potential is generated there, sooner if the myelin were not present. Moreover, ion cross the membrane only at the nodes of Ranvier, because other area which is covered by the myelin, that is showing the insulating area of the membrane. So the myelinated axons are more cost effective than the unmyelinated one, and myelin also adds efficiency in speed and metabolic cost, and it saves neurons in the nervous system, because the axon can be thinner. So that's how we can say that myelinated axon, they are more cost effective, and they will propagate the action potential more rapidly as compared to the non-myelinated axon, because in case of non-myelinated axon, there will be leakage of the charges. Conduction velocities range from 0.5 meter per second in case or 1 mile per hour for small diameter unmyelinated fiber. If the axon fiber is unmyelinated, the conduction velocity will range from 0.5 meter per second or 1 mile per hour. And if the myelin is having, if the fiber is having large diameter, and that is fiber, the conduction velocity may increase up to 100 mile per second, 100 meter per second, or 225 mile per hour. So this is the difference between the small diameter fiber and unmyelinated fiber to that of the large diameter fiber and myelinated fiber. At 0.5 meter per second, an action potential, it will travel in the distance from the head to the toe of an average size person in about 4 seconds. Mean if we have a person whose height is average mean, that is maybe 5.5, 5.6, this is the average height. In that particular case, the action potential will travel from the head region of that 5.5 feet high person to toe of that person in about 4 seconds. And the velocity of this action potential propagation is 100 meter per second, and it takes about 0.02 seconds. So it takes about 2 seconds, 0. Sorry. So that is in case of if there is myelination, it will reach about 4 second velocity of 100 meter per second. So there is current, how the current is generated in a myelinated and an unmyelinated axon. Here you can see that this is the myelinated axon, and here are the site or gaps which are called nodes of Renwire. Here the action potential is actually generated, and here you can see that positive ions, they are moving towards the negative ion. In case of non-myelinated fiber, this is the site of the action potential, and here you can see that action potential is generated at very slow pace, at very slow rate. So how the action potential is initiated? The action potential, it is basically initiated by the stimuli. So we can say that stimuli are the initiator of the action potential, and action potential are actually initiated in various types of the neurons. For example, in afferent neurons, the initial depolarization to the threshold is achieved by a graded potential. I have told you when we have discussed the graded potential that all types of the potential initially they are called graded potential, and if that initial potential has reached up to threshold value, that potential will be converted into action potential. So when we talk about the afferent neurons, as I have told you that afferent neurons are having sensory receptors at their ending, which are specialized areas for the change in the external environment or any stimuli. So initially there is graded potential at the receptors of the afferent neurons, and this can also be called as receptor potential, which is actually generated in the sensory receptor, and this potential is of the graded nature. And these are the ends which are far away from the central nervous system, and where the nervous system is actually encountering the external environment, but these areas are far away from the action from the central nervous system. In all other neurons, the depolarization to threshold is due to the graded potential generated by the synaptic input to the neurons, or to a spontaneous change in the neuron membrane potential, which is also called as pacemaker potential. All of you know about the pacemaker wall, which is also called as pacemaker SA node, and SA wall is called pacemaker wall. I mean, it is basically the spontaneous change which take place in the body, and it also take place in the potential which also take place in the neurons. So when this spontaneous potential is generated in the membrane of a neuron, that is called pacemaker potential. And spontaneous generation of the pacemaker potential occurs in the absence of any external stimuli, and it is basically an inherent property or inbuilt property of the certain neurons that they can even produce a potential even in the absence of the stimuli, even without the application of the stimuli. So that is called spontaneous potential or the pacemaker potential. And as all of you know that our smooth muscles of certain visceral organs, and even our cardiac muscles, they are showing the spontaneous potential. I mean, they are our cardiac muscles, they are showing involuntary movement or involuntary contraction and relaxation, even without the application of any stimuli. So in these cells, the activity of different types of the ion channel in the plasma membrane, initially that causes a graded potential, and that graded potential is called pacemaker potential. So if the threshold is reached, as I have told you that all types of the potential are initially of the graded nature, but once the threshold is reached, that graded potential will be converted into the action potential. So if the threshold is reached, an action potential will be generated, and membrane will repolarize, and it will begin to depolarize, or it will start to produce the action potential. So there is no stable resting membrane potential in such cells because of the continuous changes in the membrane permeability, and the rate at which the membrane depolarized to the threshold, it actually determines the action potential frequency, that how quickly the membrane is reaching up to the threshold value, and it is generating the action potential. Pacemaker potentials are very much important in certain rhythmical behavior, such as the breathing, the heartbeat, movement within the walls of the stomach and intestine. These are controlled by the pacemaker potential. So here you can see that these are the involuntary potential which are produced by certain cells of the smooth muscles, of the cardiac muscles, and that is called pacemaker potential. But once it reaches up to the threshold value, which is -55 millivolt, it will be converted into action potential. So this is the difference between the gradient potential and action potential. That gradient potential, the amplitude or intensity of the gradient potential varies with the initiating event. When we move away from the initial site of stimulus, the magnitude or intensity of the gradient potential will start to decrease because it is propagated in a decremental fashion. But action potential are generated in all or none once the membrane is depolarized to threshold amplitude. Now the intensity is independent of the stimulus strength. Now it will be independently propagated all along the length. It can be sum up, but it cannot be sum up. The action potential cannot be sum up, but this gradient potential, it can be sum. It has no threshold value, but action potential is having the threshold value, which is about 15 millivolt less negative than that of the resting membrane potential. Gradient potential has no refractory period, but action potential has refractory period. It is either having the absolute refractory period in which no further stimuli can be applied, and it is also having a relative refractory period, which is a short period of time in which you can apply a second stimuli as well. It is conducted decrementally. That amplitude of the gradient potential will decrease with the distance, whereas it is conducted without decrement. The depolarization is amplified to a constant value at each point along the membrane. The duration of the gradient potential varies with the initiating condition, whereas the duration of the action potential will remain constant for a given cell type under constant condition. It can be depolarized or hyperpolarized. Action potential mostly is a depolarization. Sometimes it can overshoot, but most of the time action potential produces the depolarization of the membrane. It is initiated by the environmental stimuli, for example, receptors by the neurotransmitter or the synapse, or spontaneously. But action potential is always initiated by the gradient potential. So it means that all type of the potential which are generated by a membrane, initially they are of gradient nature. Once they reach up to the threshold value, they will be converted into the action potential. The mechanism of the action potential, gradient potential, it depends upon the lesion sensitive channel or other chemical or physical changes, whereas the mechanism of the action potential, it depends upon the voltage gated channel, which are either sodium channel or potassium channel. So this is all about the all are non-principal and action potential as well as the gradient potential. If you have any question, you can ask. कोई क्वेश्चन नहीं? समझ आ गई सब लोगों को? ये समझ आ गई आप लोगों को? Are you there? Okay.
Yes, sir.